Okay, so we are now going to look into evolution and what are the kind of inputs that lead us to end up with this huge family tree of the animals. So many different kinds of animals. And those are just, we're just thinking about the animals that are living currently on the earth, not to mention all the ones that have long ago gone extinct. So we have to look into the process of evolution as our explanation for how did we get all these different kinds of animals. And we're going to approach it from a particular angle where we're not going to be too focused on exactly kind of the traditional model, if you've taken a bio course, of like exactly how does DNA get turned into a protein and things like that, and how DNA mutation leads to particular consequences. We're going to kind of look at it from the opposite angle. We're going to look at proteins first. Another way to think of proteins in the context that we mean it is that proteins do all the work in a cell, in a body, in an animal. That's overstating it a bit. There's other kinds of molecules that are important, but we kind of think of proteins as little tiny, tiny machines that all have different jobs. And so if we're able to zoom way in, if we were able to drop ourselves inside a cell or into the bloodstream, we'd be able to look around and see a scene that looks a bit like this very busy diagram, right? So we would see in a living cell, in a living animal, all kinds of tiny, tiny molecular machines at work. And these machines each have very specific jobs, locations, behaviors. So we can see here, I'm just going to call up a few. Some of those proteins, some of those molecular machines are very small. Others, like this big, tall, tube-looking structure, are gigantic. They can't even fit on this animation or on this poster. They extend far off of it. Some of these proteins stay in one place for their entire existence. Some are cruising around the body constantly in motion. We can see on this little kind of like zigzaggy structure in here, that is representing a cell membrane. So some of our little molecular machines are embedded in the membrane. They kind of stick out on either side of the membrane. They all have different shapes. Inside the cell, outside the cell, there's a bunch of proteins. Like this one has a kind of a cool flower shape. This one's like more of a triangle. So they have different sizes, different shapes, different jobs. Some of them are running around trying to make sure chemical reactions happen and the creature stays alive. Some of them, like the ones here, this section here, these are the proteins in your muscles that allow your muscles to contract. Some of your little protein machines are over here working away at DNA to do some sort of DNA job. Some of them seem to be wrapping up the DNA. Some of them seem to be peeling DNA strands apart from each other. So we have all these busy, busy, busy molecular machines. And I'm going to give you the link to the actual interactive here so you can kind of play around with this image and zoom in and find out what are all these different crazy shaped machines doing. So we've got that result. We know this much about cells in humans, in other animals, even in plants. Plants also have all these little molecular machines. So we know we have tons of different protein, tiny machines doing their jobs, keeping us alive every second of every day. So what we want to find out is, how did we get all these machines? They all are different shapes, sizes, different jobs, different capacities, different purposes. And so there have to be, in order to make any machine, just like if you wanted to build a car engine, if you wanted to build a computer, if you wanted to build an IKEA cabinet, you need some instructions to do that, right? So you have to have some guidance on like, how do I put this together? Which part goes with which? If you've ever put together an IKEA cabinet, you know that things can go quite terribly wrong. So we're going to focus in on the actual manufacture of those protein machines. So each one of those proteins, with all their different shapes and sizes, had to get built. And in order to build it, the cell that built it needed to have some instructions for how it gets put together. So if we look at where the instructions are, we're zooming in on the cell nucleus over here, and we find our DNA. And that is the instructions for making whatever protein machine you're trying to make. You have tons and tons of DNA, all animals do. And so different sections of that DNA are going to hold the instructions for making specific proteins. So, and your body doesn't need to make every single kind of machine every moment of the day. So there's a mechanism for the body to kind of demand a particular kind of machine that it needs. Like, oh, we need to ramp up production of this kind of machine that moves things around the body for whatever reason. So you would just be going to that particular section of the DNA, kind of going through the file cabinet and being like, oh, here it is. Here are the instructions for making that specific kind of protein machine. And then the trick is, in order to make the actual protein machine, that manufacturing process happens out here in the cytoplasm. So we can see this little kind of balloon shaped thingy here. This is called a ribosome, and its job is to make proteins. So it's actually manufacturing the proteins. The instructions for how to make that protein are lying in the nucleus, and they never come out of the nucleus. So we have this little in-between molecule called an mRNA, and it's going to carry the instructions. You can see it's kind of escaping through this little pore in the nucleus. So now we've made a copy of the instructions for the particular protein we want to make. And that copy, that mRNA, is being sent out into the cytoplasm and shown to the ribosome. So we have mRNA labeled here. So now we're zooming in on that copy of the instructions. mRNA comes out into the cytoplasm, is like, here you go. I was told to go get the instructions for making this particular kind of protein. Here they are. I went to DNA. I copied down what the DNA told me, and here it is. Here's the copy of the instructions. So the ribosome will say, thank you very much. It'll grab onto those instructions, and it'll start reading them and figuring out, just like if you read instructions for putting together an IKEA cabinet, you read the instructions, and then you actually go do the thing. The ribosome is the thing in the cell that reads the instructions and goes, oh, okay, based on these instructions, I'm going to build this. So what we see here, what the ribosome is actually building, looks like this candy necklace kind of thing here, and it's labeled growing polypeptide chain. Fancy way of saying baby protein just getting built. Now, all those colors on the beads, that is meaningful because the building blocks of protein are these kind of little, we often represent them as if they're beads. They're much more complicated than that. But the different colors represent different specific amino acids. That's what the building blocks of protein are. You know, if you pay attention to something like nutrition, people are often talking about aminos. What amino acids are you getting in your food? So each one of these little beads and the different colors, these are our amino acids. And the order of those amino acids and how many they are is critically important to what kind of molecular machine you're building. So the mRNA is what tells the ribosome, okay, this is what the protein needs to be. It needs to be this particular sequence of amino acids, right? It needs to be the right order. It needs to be the right number of each kind of amino acid. The entire protein needs to be a certain length. All of that information is what the mRNA is carrying. And then the ribosome has this cool trick of being able to read the mRNA's instructions and turn that into a string of amino acids. So what you can see here, we've got this word that says codons. The ribosome reads that mRNA in 3-letter chunks. mRNA speaks this letter language, this what we call nucleotide bases. And they come in 3-letter chunks reliably. So each 3-letter chunk is called a codon. And the way that gets translated into a protein is happening right here in the ribosome. You can see there are these tRNA molecules, and they're the actual translator. So they're going to read a specific sequence on the mRNA, and they're going to be like, oh, I recognize that sequence. When I see GCA, that means I need to bring over this specific kind of amino acid. A different sequence of 3 letters, so we can see here, the next codon here, GAA, different color bead. So that means that this tRNA is like, oh, when I see GAA, I know to bring over this particular amino acid. So that keeps happening as the ribosome chugs its little way along that mRNA molecule, reading those instructions and saying, oh, okay, when I see that particular 3-letter code, it means I add this particular amino acid. So it's almost like if these machines were thinking, it's almost like they're using a dictionary, right? So this is, if we imagined that they are literally understanding what they're doing, you can kind of picture the ribosome, the tRNA, going over to a dictionary sort of like this and trying to figure out, okay, what we're trying to do is look at the original instructions from the DNA. We have this little go-between molecule called mRNA that's bringing those instructions out to the ribosome. And then the ribosome is like, all right, when I see these 3 letters, I attach this particular amino acid, and that's how I know how to construct this particular molecular machine. So What we want to look at here is if we were to go back into the nucleus and drop in on DNA and say, all right, DNA, you never get to leave the nucleus, but you're the one that holds all the instructions for how to build things. So I want to look at the original letters in my DNA molecule and say, okay, once this whole process runs, what is that particular letter sequence going to actually mean in terms of an amino acid that I'm going to stick onto my growing protein molecule? So, for instance, you can see how this is constructed. So we're always working with these 3-letter chunks, these codons, right? So the way to read this kind of wacky looking dictionary is we've got this little corner. Oop, take my drawing tool. This says first base, not in like a baseball sense, but saying, okay, if you've got a codon, right? So we've got a codon is going to be 3 letters. So this will be the first base, then there'll be a second base in this position, and then a third base. So the way to read the dictionary is to say, okay, over where it says first base, in that column, I've got 4 options, right, for what my DNA might say. So my first position, let's say it's A, all right? So I went to the DNA, I zoomed in on a particular spot in the DNA, and I read, okay, this DNA says A for the first base. All right, now I'm going to keep moving down my DNA molecule and say, okay, what letter comes next? So now I'm going to move over to this second base, right? And see, okay, well, what's in that position? All right, I keep reading my DNA from left to right. All right, let's say that the second base is a C, right? So now I've got C in my second position. All right, now I hop way over to the right side. Here's the third base. All right, I'm reading my DNA molecule. I see, okay, there was A first and then C, and then my third letter position is a G. So this is my original DNA instructions, right? I looked at a literal DNA molecule and I see that the 3 letters in the spot that I'm staring at say A, C, G. All right, so if those instructions were then brought out into the cytoplasm, handed to the ribosome would latch onto that and be like, thanks for bringing me the instructions. I'm going to get to work on building. And it's going to look at those 3 letters and say, okay, A, C, G. All right, so my first base was an A, right? So now I know I'm somewhere in that range. My second base was a C, and my third base was a G. All right, so if my first one was an A, I'm looking at this particular, so I got a little bit of an unsteady drawing hand here. All right, so I'm thinking, okay, this is where I'm at first. And now, all right, I'm looking and saying my second base is a C, so that means I'm going to be zooming in on this column. It's good to not have too much coffee before doing this. Okay, so now I have A, C are my first ones, and then I've got G. Okay, so I'm zooming in on this particular box, right? So now my overlap is in this box because these are all the possibilities so far, and you can see, I'm just going to erase a little bit of that. Boop. All right, so we can see that sure enough, all of these options in this column all start with A, C. So now I'm looking over here and saying, okay, my third position is a G. So what I'm looking at specifically is this little box right here, right? So my DNA, my original DNA sequence was ACG. But what I want to know, if I want to actually try to make a protein here, make a molecular machine, I need to know, all right, when the ribosome grabs onto those instructions and it reads ACG, what does that tell it in terms of which amino acid to kind of go grab off the shelf and stick onto this thing? So that's what this box is, right? So this green thing here, this is showing us that ACG is going to is going to code for the specific amino acid threonine, right? So anytime the DNA instructions, the original instructions, read the sequence, the codon ACG, that's going to mean you're adding a threonine amino acid to your protein chain. Now, what you'll probably start to notice here is that there's more than one way to code for a particular amino acid, right? So if we were to take, let me clear off this stuff so we can clear this out. What we know from that example is that if I have ACG, this is going to lead to a threonine, right? So, but what you see when you zoom in on the threonine box is that there's a lot of other codons that would also code for threonine, that would also tell the machinery in the cell, okay, it's time to add a threonine. Not only ACG, but also, let's see, what else? We've got ACA, ACC, ACT, all of those. We've got 4 different codons that all make threonine, right? So there's not just one, it's not a one-to-one kind of thing. Now you'll see if you just kind of scan over all the different kind of color blocks and let your eyeballs take this in, that some amino acids, there's like a lot of different ways to make them. So if we look over here at, say, leucine, that amino acid, there's a lot of different codons that can tell the cell, add a leucine here, right? So if the original DNA instructions say TTA, TTG, CTT, CTC, CTA, or CTG, every single one of those codons is going to tell the cell, add a leucine amino acid at this spot. So that means there's a lot of different ways to arrive at the same amino acid. And we're gonna play around with this. It's kind of just feels like sort of like, okay, that's nice. I don't see what impact that necessarily has on things. So we're gonna play around with a few different possible examples that are gonna tell us, does that matter if I have a lot of different ways to kind of call for a specific amino acid? Do I care? Does that impact the functioning of the cell? Does it impact the animal? So we're going to go down to, we're going to take a look at those amino acids. So I'm just orienting you to a few different things to think about so that you can proceed with the worksheet that accompanies this particular exercise. So we were looking at that sort of dictionary setup that just had the names of amino acids, and we see the names here. So if we, kind of look at a few different places here, we've got, so here's, whoop, didn't mean to do that. So I can see we've got threonine. There's that threonine we were talking about. So it actually is a physical thing. Threonine, so all the amino acids are these little molecules. And each amino acid has its own name. So you can see the names are all listed here. So histidine is one amino acid, arginine, lysine, glutamate. They have all these different names. Generally, the number that's given is that there's 20 amino acids. It's fine for us to use that number. And you can see that there's some commonalities to them all. So you can see sort of like the upper part of an amino acid. That's pretty much the same. If you look across all these amino acids, that top section basically looks the same. There's a couple exceptions that you can probably start picking out on your own. But that's the basic backbone of every amino acid. Most of the time, we're not super interested in that part of the amino acid. We're more interested in the parts that are in these blue boxes hanging down, because that's the part that sets the amino acids apart from each other. That's what makes them different from each other. And those can be very different from each other. So we've got these kind of funky ones that have these big, fancy chemistry looking rings. We've got one case, at least, with the amino acid glycine, where what's in that blue box is just this one lonely little hydrogen. So one thing to just kind of take in, first of all, is just think about that these are physical structures. They are so small that we cannot see them, but they are physical 3D structures that exist in a cell. And we know that, in the real world, like in stuff that you can see, we are concerned with, is something big or small? What is it shaped like? So we have to concern ourselves with exactly that with amino acids, even though we can't literally see them. So what we know is that, like, if you look across here, you can start to figure out, and we're going to play around with this with some of our activities, some of these amino acids are pretty big. I mean, they're all super tiny small, but compared to each other, some of them are very, very tiny. Some of them are big and bulky. So that's kind of the first thing for us to ask, is just sort of keeping in mind, like, what's the overall size of a particular amino acid? Is it a tiny amino acid? Is it a big, bulky one? So you kind of want to start making a mental framework for how they can really vary, and sort of sort through these mentally and say which ones look big and bulky, which ones look much smaller, because that's going to matter in terms of how they function in the protein. Don't have to know why yet. We're just taking in, yeah, all these amino acids have different sizes. Now, the other thing we want to focus in on is these kind of groupings, right? So some of these amino acids are in this box, and then there's another box down here, and there's another box here. So they seem to be organized into categories. And if we look over at the kind of labels in the sides of these boxes, we see that we're being told the different categories they can fall into, right? So these don't have to, these words can kind of wash past you for the moment. I'm going to help you sort through what's actually important for you to be tracking. So we can see here the label on this first kind of box of amino acids is saying, oh, all right, in the top left box of amino acids, all of those amino acids are these things. And again, some of these words may make sense to you, some not. But it's telling us every amino acid in that box is nonpolar aliphatic R groups. Now, you may not know anything about what aliphatic R groups are, and I'm not actually going to be concerned with that too much right now. The R group, so we should actually define that. So this is an R group. So the blue boxes represent the R groups. And the things in the blue boxes, are what make the amino acids different from each other, right? So we've got some of those ones that have the big weird kind of rings and stuff. But each one of those is an R group. Okay, so the blue boxes, I'm just going to put blue boxes. The blue boxes are what represents the R group. So every single amino acid has its own R group, and their R groups are what make them different from each other. So whenever you see R group, we're kind of picturing the blue section, sort of the part in the blue box that makes glycine different from isoleucine, different from threonine. That's what sets them away from each other. So now that we know what R groups are, we're going to focus in on what these boxes are telling us. So we've got one option is we've got this non-polar. Let me just clean this up. Okay, so we've got a couple of different options here, right? So we've got a box of amino acids that are being labeled non-polar. Then we've got a box of amino acids that are being labeled as polar. Then we've got a box of amino acids being labeled positively charged, another collection that are negatively charged, and another collection that are non-polar. So for our purposes, we're going to try to keep this very simple. We're going to use just a general model of almost simple magnetism. You may have had much more biology background than I'm indicating, but I'm just kind of assuming that we're all approaching this without a whole lot of biology background. So we're going to focus in on the fact, and this is probably something that you do know, opposites attract, right? So a positive and a negative are going to be drawn towards each other. Now, when we're looking at something like, and then I'm just going to make a messy diagram here. I'm going to put this because I'm just trying to make a reminder. So positives and negatives are always going to attract. The stronger the positive and the stronger the negative, the more powerfully they're going to attach to each other. It's kind of like imagining a big, strong magnet, a big, strong set of magnets, like really snapping together, like you wouldn't want your fingers in between there, versus some kind of weak magnets that are just like not that scary. So that's what we're looking at. And we're going to imagine, and you may know more than this about this idea. We're going to use polar to just indicate weakly magnetic. Magnetic. All right, so if you, and then non-polar, let me erase this again. So polar is going to equal weakly magnetic. Non-polar is going to represent not magnetic at all. So it's almost like if you were playing around with magnets, you'd be like, okay, I have my really strong set of magnets that are kind of intimidating. Then I've got my weaker magnets. And then I've got some wooden blocks that are just not magnetic at all. It doesn't matter how strong a magnet you try to bring over to them. Wood is just not going to respond to a magnet. We're going to think of anything that's labeled non-polar that way. It almost acts like a block of wood when it's faced with a magnet. It has no reaction to that at all. So we've got our polar, which means kind of weak magnetism. nonpolar, which means no magnetism at all, no positive negative interaction at all. And then we've got some more, oh, and then there's another nonpolar guy there. So I'm going to just kind of thread this through and note, okay, these guys are also nonpolar. So those guys also have no magnetic interaction kind of happening, no positives and negatives interacting. So then that leaves us with positively charged R groups and negatively charged R groups. And these are going to be our ones that act strongly magnetic. There we go. Okay, so these are kind of our options for how the different amino acids are going to interact with each other. So if you've tracked down, okay, I've got a couple of polar amino acids, a couple of polar R groups. That means they're going to potentially be weakly magnetic. And we have to remember that positives and negatives attract. A couple of other rules are positives do not attract other positives, right? They hate each other. Negatives do not attract other negatives. Again, playing with magnets, if you try to put two positive ends of magnets near each other, they push away. They do not like being near each other. Two negatives will not like being near each other. Only positives and negatives are going to draw towards each other. So whenever we see either a either a polar or a charged R group, we have to then say, okay, now I know there's going to be some potential for magnetic type interactions here. I have to sort out, am I looking at two negatives that are coming near each other and they're going to hate each other and try to get away? Am I looking at two positives that'll also hate each other? Or do I have this kind of magical combination of a positive and a negative, and they're going to draw towards each other and want to get closer? So we've got some amino acids that are in that kind of magnetic, either polar or charged kind of group. And then we've got others that are non-polar, that are not going to participate in that kind of magnetism, right? So we're going to come around and we're going to do an activity, because I'm going to do, let's see if we want to go down here. Doop, doop, doop. Yeah, I'll leave that for right now. So that's going to give us a chance to play around first with a little bit of how would this impact how these different amino acids interact with each other. If you're looking at two amino acids, would you expect that based on this stuff about the R group, that those two amino acids would tend to want to get nearer to each other? Are they going to kind of magnetize toward each other? Are they going to be repelled by each other because they have some sort of charge on them and it's two positives together or two negatives together? Or are they non-polar and they're just kind of like not interested in participating in any of that kind of interaction? So that's what we're going to pick up with kind of the challenge that I'm giving you with sort of walking through this worksheet. Just looking at, all right, how would we predict that these different amino acids would interact with each other based on these ideas that they come in sort of these categories? They can either be carrying some sort of charge, whether it's a weak or a strong charge, whether they act as kind of weak or strong magnets, or if they're non-polar and they don't act magnetically at all and just have no interest in participating in that.